Okay, development of monosporic embryo sac, or it is commonly known as a polygonum type. It is also commonly known as polygonum type. Okay, it is also known as polygonum type. Okay, now here what happens, already we know that three micropylar and megaspore degenerate and only one towards chalazal and megaspore become functional. So what we can say, in majority of plants, in majority of plants, three megaspore micropylar end becomes degenerate, and only one megaspore chalazal end becomes functional. Okay, becomes what? Functional. This line you have to remember. Which side? So only one megaspore towards chalazal end becomes functional. Okay. Now, this type of embryo sac formation, from single megaspore is known as monosporic development. This type of megaspore formation from single embryo sac development is known as monosporic embryo sac. Okay, so it is known as what? Monosporic. Now, here what happens? Three mitotic successive divisions occur. So one developed, one mitotic division, what happens? This converted into two. Second mitotic division, so these two converted into 4. Then third mitotic division, this converted into 8. So 1, 2, 3, 1 central, 5, and 1 here 8. Okay, so this is 8-celled stage. Okay, so it is what? 7-celled stage and 8 nucleate stage. 8 nucleate stage. Now, these are further developing. So you have to remember this. How many mitotic divisions? 1, 2, and 3rd. 3 mitotic divisions are there. So in 3 mitotic divisions, what is formation occurs? This is which side? Micropilar end. This is which side? Chalazal end. Okay. This is what? This is egg cell. These two are what? These are synergids. So how many synergids? One. One this and one this. So two synergids are there. One egg cell are there. Okay. So 1 plus 2. It is making what? Egg apparatus. So it is what? Three-celled stage. Okay. Now, here synergids contain finger-like structure. These synergids contain finger-like structure that is known as a filiform apparatus. Filiform apparatus. This known as central cell that contain n plus n nucleus type. Okay, n plus n, it is not 2n, these two n plus n existing. Okay, next these are antipodal cell. Antipodal cell. Here how many? 1, 2, 3. So 3 are there. Okay, 3 antipodal cells. Now here questions are asking that this is the figure of mature embryo sac. Mature embryo sac. Here questions are asking, this figure will give and ask mature embryo sac contained how many nucleus and how many celled structure. So you just tell me how many nucleus are there and how many celled structure are there. Yes. So what is what? 1 nucleus, 2 nucleus, 3 nucleus, 4 nucleus, 5 nucleus, 6 nucleus, 7 and 8. So how many nucleated structure? 8. And cell, 1 cell, 2, 3 cell. This is central cell. So 4, 5, 6, 7. So it is 7. So what is we can say? A mature embryo sac is 7-celled and 8 nucleate structure. This line is very, very important. What I told, a mature embryo sac is 7-celled and 8 nucleate structure. Understood what I told? Anybody? Okay. Now we will come on one by one. What is antipodal cell? Egg synergids. Now here you have to remember also that egg apparatus contains what are the components. So it is 3-celled stage or 3-celled stage structure that contain 1 egg cell and 2 synergids. Okay. Every time egg you can see here, so egg is what? Bigger. Egg cell is larger in size compared to others. Okay, other cells, egg cell is larger in size. Next we have to see organization of embryo sac, means what are the components that this embryo sac contain. Organization of embryo sac. So first what we seen? First we seen synergid cell or helper cell or cooperative cell. So first is synergid cell or helper cell or cooperative cell. Okay, synergid cell or helper cell or cooperative cell. Okay, now what they contain? Mostly they contain micropylar nucleus. Where this, in which side they are present? Towards micropyles. What we can say? This generally possess, these generally possess micropylar nucleus. Micropylar nucleus. And what they are doing? They having presence of filiform apparatus and containing filiform apparatus. Okay, they are containing what? Filiform apparatus. Now, this filiform apparatus doing what? They help in directing pollen tube. So what we can say? Which helps in directing pollen tubes in proper way. Okay, so this filiform apparatus helps in what? Directing pollen tubes in proper way. Okay, now next what we seen? Next is egg cell. Next is egg cell. So egg cell is what? It is larger than synergids. Already we seen. It is larger than synergids. It is larger than what? Synergids. And what happens? Which represent a female gamete. Which represent female gamete. Okay, so which one represent female gamete? So that is egg cell represent female gamete, and it forms what? They form, it forms egg apparatus. Egg apparatus along with two synergids and one egg cell. So this you have to remember, two synergids and one egg cell form what? They are forming egg apparatus. So synergid is there, egg is there. Next is antipodal or vegetative cell. Antipodals or vegetative cell. It is found where? Towards chalazal gland. It is found towards chalazal end. Okay, it is found where? Towards chalazal end. Okay, and these are three in numbers. Three already we seen, which are three in number, which become degenerate degenerate. When? During maturity. Okay, when maturity occurs, then what happens? This become degenerate. Now, last is what? Central cell. Central cell is there. Last one, central cell. So it is what? It is largest cell of embryo sac. It is largest cell of embryo sac. This you have to remember. This is what? Largest cell of embryo sac, which contain two polar nuclei. So, which contain two polar nuclei. How much nucleus they are containing? Two polar nuclei containing. Okay, understood? Now, so these are some, what is that? These are some mature embryo sac components are there. Okay, next we will see embryo sac having types, that we will see in next class. Okay, thank you. Hello, sir. Okay, sir. Thank you, sir.